Hi y'all, I'm going to try to do this one outside if the road noise here doesn't disrupt it too much. So 14.1, human chromosomes. Okay, so if you had to pick an ideal organism for study of genetics, would you choose one that produced lots of offspring, or one that was easy to grow in the lab, or had a short lifespan that allowed you to do several crosses per month? Well, you certainly would not choose an organism that produced very few offspring, had a long lifespan, and could not be grown in a lab. Yet when we study human genetics, this is exactly the sort of organisms that we deal with. So given all of these difficulties, it may seem a wonder that we know as much about genetics as we do today. So what is a karyotype? And that's K-A-R-Y-O-T-Y-P-E. A karyotype shows the complete diploid set of chromosomes grouped together in pairs, arranged in order of decreasing size. So to find what makes us uniquely human, we have to explore the human genome. A genome is a full set of genetic information that an organism carries in its DNA. So a study of any genome starts with the chromosomes, the bundles of DNA and protein found in the nuclei of the eukaryotic cells. To see human chromosomes clearly, cell biologists photograph cells in mitosis when the chromosomes are fully condensed and easy to view. So scientists then cut out the chromosomes from the photographs and arrange them in a picture known as a karyotype. It shows the complete diploid set of chromosomes grouped together in pairs, arranged in the order of decreasing size. A karyotype from a typical human, which contains 46 chromosomes, is arranged in 23 pairs. Y'all didn't know it, but if we were still in class, one of those crazy projects I would make you do is we were all going to go to the gym. I was going to bring my selfie stick, climb way up on the bleachers, and I was going to make each one of you do, with your body on the floor, one of the chromosomes, and I was going to take a picture of it. So, sex chromosomes. See what y'all missed. We could have done that. 2 of the 46 chromosomes in the human genome are known as sex chromosomes because they determine an individual's sex. So females have 2 copies of the X chromosome, while males have 1 X and 1 Y chromosome. Okay, Punnett squares, and y'all know what these are. We talked about these before. This is a square that illustrates why females are born in roughly a 50-50 ratio. There's a really good picture of this in your book. A human egg cell carries the X chromosome, 23 and X. However, half of all the sperm cells carry an X chromosome, 23X, and half carry a Y chromosome, 23Y. So this ensures that just about half the zygotes will be males and about half will be female. Okay, so more than 1,200 genes are found on the X chromosome, some of which are shown in a picture that you have in your book. It's got some little striated lines on there for you. The human Y chromosome is much smaller. The human Y chromosome is much smaller than the X chromosome, and it contains only about 140 genes, most of which are associated with male sex determination and sperm development. Okay, autosomal chromosomes. The remaining 44 human chromosomes are known as autosomal chromosomes or autosomes. The complete human genome consists of 46 chromosomes, including 44 autosomes and 2 sex chromosomes. So to quickly summarize, the total number of chromosomes present in a human cell, biologists write, is 46, XX for females and 46, XY for males. So what patterns of inheritance do human traits follow? Many human traits follow a pattern of simple dominance. The alleles for many human genes display codominance inheritance. And what patterns of inheritance do human traits follow? Because the X and the Y chromosome determine the sex, the genes located on them show a pattern of inheritance, which is called sex-linked. So many human traits follow a pattern of simple dominance. For example, a gene known as MC1R determines the skin and hair color. Some of the MC1R's recessive alleles produce red hair. An individual with red hair usually has 2 sets of these recessive alleles, inheriting a copy from each parent. Dominant alleles for the MC1R gene help produce darker hair colors. So another trait that displays simple dominance is the rhesus, or the Rh blood group. The allele for Rh factor comes in 2 forms, the Rh positive and the Rh negative. Rh positive is dominant, so an individual with both Rh positive over Rh negative is said to have Rh positive blood. And the Rh negative blood is found in individuals with 2 recessive alleles. So the alleles for many human genes display codominant inheritance. One example is the ABO blood group, determined by a gene with 3 alleles. Okay, you will have a table in your book or your handout that shows the codominant and the multiple alleles for the blood groups. And the table in there shows the relationship between the genotype and the phenotype for the ABO blood group. Remember, phenotype is what it looks like. Genotype is what it sounds like, the genes that are involved. So it also shows which blood types can safely be transfused into people with other blood types. Before we started the class, I put a really neat little blood donor post on your Facebook page for you. If a patient has AB negative blood, it means the individual has I to the A and I to the B alleles for the ABO gene, with 2 Rh negative alleles from the Rh gene. And if a patient has AB negative blood, it means the individual has I to the A and I to the B from the alleles of ABO gene with the Rh negative alleles from the gene type. That'll make much more sense if you just look at the chart that's in your book. Alleles for I to the A, I to the B are codominant. They produce molecules known as antigens on the surface of red blood cells. And individuals with the alleles of I to the A and I to the B produce both A and B antigens, making them type AB. The I allele is recessive. Individuals with alleles of IA, or the dominant IA, small a, produce only the A antigen, making them type A. Those with B, it's the same thing. If you've got 2 of the dominant B and then 1 recessive, that's going to get you type B. The homozygous for the allele, the I allele II, produce no antigen or are said to have type 0 blood. And that's 2 small i's. Okay, the genes located on the X and Y chromosomes show a pattern of inheritance called sex-linked. And a sex-linked gene is a gene located on a sex chromosome. So genes on the Y chromosome are found only in males and are passed directly from father to son. And genes located on the X chromosome are found in both sexes, but the fact that men have just 1 chromosome leads to some interesting consequences. So, for example, humans have 3 genes responsible for color vision, all located on the X chromosome. In males, a defective allele for any of these genes results in color blindness, an inability to distinguish certain colors. The most common form is the red-green color blindness, and it occurs in about 1 in 12 males. Among females, however, color blindness affects only 1 in 200. In order for a recessive allele like color blindness to be expressed in females, it must be present in 2 copies, 1 on each of the X chromosomes. And the recessive phenotype of a sex-linked genetic order tends to be much more common among males than females. So most of the time it's your males that are colorblind. Okay, X chromosome inactivation. If 1 X chromosome is enough for cells in males, how does the cell adjust to the extra X chromosome in female cells? Well, in female cells, most of the genes in one of the X chromosomes are randomly switched off, forming a dense region in the nucleus known as a bar body. Bar bodies are generally not found in males because their single X chromosome is still active. So X chromosome inactivation also happens in other mammals. So in cats, a gene that controls the color of coat spots is located on the X chromosome. So one X chromosome may have an allele for orange spots, and the other X chromosome may have an allele for black spots. In cells in some parts of the body, one chromosome is switched off. In other parts of the body, the other X chromosome is switched off. So as a result, the cat's fur has a mixture of orange and black spots. So male cats, which have just one X chromosome, can have spots of only one color. If a cat's fur has three colors, white, orange, and black spots, for example, you can almost be certain that the cat is female. So how can pedigrees be used to analyze human inheritance? The information gained from pedigree analysis makes it possible to determine the nature of genes and alleles associated with inherited human traits. So to analyze the pattern of inheritance followed by a particular trait, you can use a chart called a pedigree, which shows the relationship within a family. A pedigree shows the presence or absence of a trait according to the relationship between parents, siblings, and offspring. There is a diagram in your book, and it shows what a pedigree looks like. It's white, has white circles, blue circles, and squares, and basically it looks kind of like a family tree. So I'll need you to take a look at that on your own. I'm going to read through the things about the chart, even though you can't see it right now. The pedigree shows one human trait. A white lock of hair just above the forehead passes through 3 generations of a family. The allele for the white forelock trait is dominant. At the top of the chart is a grandfather who had the white forelock trait. 2 of his 3 children inherited the trait. 3 grandchildren have the trait, but 2 do not. So because the white forelock trait is dominant, all the family members in the pedigree lacking this trait must have homogeneous recessive alleles. One of the grandfather's children lacks the white forelock trait, so the grandfather must be heterozygous for the trait. The information gained from pedigree analysis makes it possible to determine the nature of genes and alleles associated with inherited human traits. So based on a pedigree, you can often determine if an allele for a trait is dominant or recessive, autosomal, or sex-linked.